So, hello everyone and welcome. I am Alyssa Taylor Bustillo, Executive Director of the GreenTB Foundation, and I am thrilled to have you all joining today's community learning webinar on understanding biomarkers and patient samples, key factors in improving clinical trial design. So, we are so excited to have Dr. Anna Falzer from Combined Brain with us today to share her expertise. We have worked very closely with Dr. Falzer and her team, really collaborating to advance research and improve outcomes for our community. So we are very excited to have her join today. After her presentation, we will have some time for Q&A questions. So if you guys have any questions during the presentation, feel free to pop them into the chat. And if not, wait until that time towards the end to ask questions live. But with that being said, we can go ahead and get started. So, Dr. Falzer, over to you.
Sure. I'll start sharing my screen. Thank you for that really sweet introduction, Alyssa.
Sure.
Kind. And I should first say, I'm not going to say that I'm an expert in biomarkers, but that's what I've done for the last decade or so, or more than I would care to admit. But anyway, so I will talk about what I know. So I can't guarantee I can answer everybody's questions, but okay. So if I share my slideshow, does that still work?
Yeah, that works perfectly.
Yay! Okay, cool. All right. So you can see I sort of stole some of your honeycomb here, but that's such a cool color. Okay. So for whatever time we have today, like Alyssa said, I am going to talk a little bit about the role of biomarkers in the drug development process. And this is particularly important, as you just said, Alyssa, for rare diseases. And, I don't want to assume people know what I am talking about. So I want to make sure I do justice to talking about what is a biomarker. I feel like everybody throws the term around, and even myself, I sort of get a little confused sometimes, but it seems really important. And so I want to make sure everybody really understands what a biomarker is and why lots of scientists and clinicians are obsessed with the concept of coming up with a biomarker for GreenTB related disorders. And the second thing is really, why are they important? What is the role of biomarkers in the overall drug development process? What are examples of biomarkers that have been used in other neurological disorders? Biomarker data, potential biomarker data in GreenTB patient samples, and then how can patients and their families get involved in biomarker development? And then I put obviously time for questions and answers, if there are any. Okay, so this is a little bit of a generality, but biomarkers, the way they distinguish themselves from surveys or other sort of caregiver or patient-reported measures is that biomarkers are objective measures, which can be used to predict disease status, progression, and or even how well an individual is going to respond to a particular treatment. The really interesting, important thing here is that we use the term objective, right? It's not based upon how you feel or how your parent or caregiver thinks you feel. It's an objective measure. These are really important to include in clinical trials, particularly when you're looking at the effect of a particular intervention, because this is objective. There is no subjective part of this to determine whether something is actually getting better or getting worse. So what are examples of different types of things that you find biomarkers in? And I have put them into two basic categories right now, although this is certainly not exhaustive. So the first category would be biologic biomarkers. So these would be things like mRNAs or genes, as you guys are probably very familiar with. There can be epigenetic biomarkers, and I'll talk a little bit more about what epigenetic means, but it basically means modifications to a person's genome. Biomarkers can also be proteins or metabolites or even different species of microbiota. And where you find these biomarkers, different fluids, different tissue types, different cell types, etc., that is also variable. So this list is not exhaustive at all, but tend to be some of the most common specimens that you see biomarkers identified in. And those would be cerebrospinal fluid, which would be the fluid that sort of bathes the brain. Blood, urine, saliva, and even stool can be used to identify biomarkers. The other big category here, and this is something that's really particularly relevant for disorders that have epilepsy, would be physiologic biomarkers. And one of the most common ones, again, although not exhaustive, is the use of electroencephalograms or EEGs, which I'm sure many of you are very familiar with. And then a version of this, which utilizes the EEG measuring device, is the event-related potential, or just a way someone responds to a particular stimulus. So overall, very, very generally, EEGs measure electrical activity in the brain, which I'm sure, again, many of the people watching or listening are very familiar with, unfortunately. And more importantly, alterations and patterns of this EEG can signal a seizure or a different feature of a neurological disorder. And this is an example of the cap that you use when you're doing an EEG, or the most common way to measure an EEG. Okay, so this is a schematic which the founder, Dr. Bichel, put together, but I use it almost every time I speak about biomarkers or outcome measures, is because it really nicely captures the overall drug development process for a lot of disorders, but in this case, we'll use it to talk about drug development for a rare neurological or neurodevelopmental disorder like GRIN2B related disorder. So on the left-hand side, I'm just going to assume that you guys can see my cursor here. On the left-hand side, you see the gene, the causal gene that is causing the symptoms in the infant or adolescent or young adult. It's causing the individual person to be symptomatic. So in this case, it would be GRIN2B. And then on the bottom of this figure here, you can see that the next thing that you usually have to establish or develop are rodent and cell models of the particular disorder. And this is something actually that the GRIN2B community has done a really great job of. Meaning that there are a lot of patient-derived or patient-donated cell models that have been developed that researchers can use to identify potential treatment strategies, or just basically understand the mechanics of GRIN2B pathogenic variants on a cellular system. And then usually one step up from looking at cell models is also looking at rodent models of the particular disorder. So mice, rats, etc. are really common models of uses for this that carry various GRIN2B variants that mimic the disease in certain respects. So, and I won't spend a lot of time going into all of these, but a natural history. So how does the disease change with age and potentially sex as an individual develops from infancy into adulthood? It's also really important to look at the mechanisms that are involved. So what do GRIN2B pathogenic variants do in terms of changes or disruptions to cellular biology that have those deleterious consequences on neurons and other cell types that then lead to symptoms in children. Moving down, further down on the right, you can see the disease concept model or disease burden. These are objective ways to measure the symptoms, the variety of symptoms that are present in GRIN2B related disorder patients. So what are the different parts about GRIN2B that impact patients and families? And this study is really critical, and this is also one that is directly tied to community involvement. The symptoms that are reported here through caregiver interviews, these are the symptoms that should be used as outcome measures for clinical research or potentially clinical trials in GRIN2B related disorder. Now, underneath this, you can see finally biomarkers, the importance of biomarkers. And we've already talked about what those are. Those are objective indicators of sort of the disease status of a particular individual, or they can also be used to predict how well they will respond to a particular experimental treatment. These, again, similar to the disease concept model, are parts of the drug development process that are absolutely critical for the community to be involved. And then moving further to the right, you can see patient-centered outcome measures. Those are, just as the name would suggest, outcome measures that are centered around things that are important to the patients and their families. And then you have FDA approval and clinical trials, and then specific drugs getting approved for GRIN2B related disorder patients. I'm going to skip right through this because I forgot that I had that slide. That sure would have been good for me to remember. Okay, so the next thing is so what are examples of biomarkers that have been used in other neurological disorders? And I pick these two here because they are certainly not specific to adult neurodegenerative processes, but have been implicated several times in the context of pediatric neurological disorders as well. So one really important thing, as I suggested before, is that biomarkers change with disease and age. That's part of what makes them biomarkers, right? They are markers of disease status. And two examples of this, which some people may be familiar with, but I just pulled out an example here from a paper that was looking at biomarkers in Alzheimer's disease. Alzheimer's disease being one of the most well-known adult neurodegenerative disorders currently in the United States. So there are two well-known biomarkers or indicators of disease status in Alzheimer's, and those are measuring the proteins known as neurofilament light, or NFL, and then tau protein levels. And in fact, we know that levels of those two proteins, both in the cerebrospinal fluid and in the plasma, increase as disease progresses. So in fact, if you look at these four figures here, two on the top, two on the bottom, the two on the top here are looking at tau levels in a particular individual. And then the bottom one on the left-hand side here is looking at neurofilament levels. And then this third one or fourth one over here is looking at a different biomarker. This is called gliofibrillary associated protein, or GFAP for short. And on the horizontal axis here, you can see age, which is being used as an indicator for disease severity. So as a patient ages with Alzheimer's, most typically they get more severe. And without going into a ton of detail on the dots and the lines, basically what I'm trying to show you is that for these three different proteins, tau, neurofilament, and GFAP, as the individual gets older, the level of these proteins increase. And so if you can imagine, if you did some sort of experimental intervention and you could level out the increase in this protein, or make it not increase with age, or even more, decrease with age, that would be a sign that a particular clinical intervention or some sort of intervention was having a beneficial effect on that person with Alzheimer's disease. Okay. So more examples. Let's see if I can use this marker. Some specific examples about physiologic biomarkers that I introduced on the last couple of slides. Like I said, one of the primary ones in the context of rare neurodevelopmental disorders, or developmental epileptic encephalopathies, so disorders associated with epilepsy. Probably the most common one are EEGs. So EEGs can be diagnostic. So you can prove, right here is sort of a snapshot of what an EEG reading looks like. And I'm sure, again, many of you are familiar with and have seen these in your own patient or family members' medical records. This is what EEG readings look like. And you can sort of see abnormal patterns here being highlighted. So these EEGs can be diagnostic. They can demonstrate when or if your child or family member is having some sort of epileptic episode. They can also be used as tools to identify biomarkers. Are there unique patterns in these EEG wavelengths or readouts that can, one, identify whether the person potentially has a GRIN2B mutation, if there was specific to the particular genetic mutation, or more likely that there's just something that's unique and novel compared to neurotypical EEGs that could be fixed or improved with a clinical intervention. And more importantly, like I just said, EEGs can be correctable. They can be fixed fairly quickly, or they're very sensitive to intervention, whereas, changes in something like cognition or behavioral symptoms might take a little bit more time to see subtle changes or improvements in us. Here is just a caricature of someone wearing an EEG cap. I had one of those on the last slide. These figures right here, these two sort of overviews looking over someone's head with the different colors here. This is an example of a different type of assay that was looking at the event-related potential or evoked-related potential. And what this measures, you can see the different colorations here. What these measure is differences in how an individual responds to a particular stimulus. So what areas of the brain are highlighted or respond very quickly or slowly in response to, for example, an image that the child sees, or a noise, an audio song, short clip being played. What are the differences in how their brain responds compared to a neurotypical individual's? Okay, so one paper that I actually pulled, I was reviewing this last week, and I thought it was really relevant for today's talk. This was actually published 1 or 2 years ago by this group, but it really is one of the potential things that could be a biomarker for GRIN2B, so I thought we should just introduce it as an example for how blood-based biomarkers might actually be very useful in the context of GRIN2B-related disorder clinical trials. So this paper, the title of the paper is Childhood Adversity Increases Methylation in the GRIN2B Gene. Now, we're not studying childhood adversity in the context of today's webinar, but the interesting thing from this paper is that GRIN2B methylation status was associated with certain clinical symptoms. So very first thing here is, what is methylation? Well, on one of the very first slides, one commonly used biomarker for these types of disorders, especially genetic disorders of genetic etiology, are epigenetic changes. And what those are small chemical modifications to the genome of a particular individual. So you guys are probably very familiar with seeing cartoons that show nucleotides or bases, G, C, T, A, etc. And this is the genome. These are the genetic basis upon which DNA, RNA is built, and then subsequently proteins. But these bases, these nucleotides, are themselves chemical structures, and so they can be modified by other chemical structures. In fact, this is sort of a zoomed-in version of what this looks like. And basically, these M's right here represent small chemical modifications that can change the way DNA is wrapped or folded or exposed, etc. And those subsequently change how much of a gene is made or how little a gene is made. And for people or parents who have children with GRIN2B related disorder, know that the level of expression and abundance of particular genes are what are associated with various clinical symptoms observed in their family member. So methylation status is associated with certain clinical symptoms. And in this case, in this paper, they found it to be really heavily associated with certain emotional problems or depression and different types of aggressive and non-aggressive behaviors. So the interesting thing about this is that methylation status can be altered, both environmentally, as well as sort of, there are being developed right now more targeted approaches to change the methylation status of specific genes. In this case, could be GRIN2B. So because of this, methylation status could potentially be a biomarker for GRIN2B related disorder. I say potentially because this certainly hasn't been validated in a population of GRIN2B individuals, but certainly there is scientific justification for looking at something like this. One thing that this would require to really better understand and explore would be to get blood samples and phenotypic information from the GRIN2B related disorder community to really validate this initial finding and see how relevant it is to the GRIN2B related disorder population. Okay. All right. So what kinds of samples can be used for biomarker research? And I apologize, my formatting is a little bit wonky, but that's okay. Everyone can still see. So there's a variety of different sample types that people have used and have seen biomarkers in. And here is a really broad, long list of potential biological samples that people have previously used to look at biomarkers. One of the most common ones is various blood components. Blood spot cards are just drops of blood. Cord blood, saliva, urine, nasal brushings, DNA from buccal swabs, even stool samples, as we said, can be used to look at different populations in microbiota. And then biomarkers can be looked at in non-CNS tissue. Another way to say that would be tissue that's outside of the central nervous system. So other peripheral tissues that we know are involved and disturbed with GRIN2B variants. So these are the types of samples that people can use to explore potential biomarkers. And one really important thing, I'm not going to go into all of these, but one really important thing to remember is that when we're looking at biological samples, it's really important to have what I'm calling phenotypic data to better interpret any results that you have from your biological samples. So it's important to have, sort of indicators of disease severity. It's important to know if the child has epilepsy or not. It's important to know if they're on any sort of diet that could modify some of the biomarkers that we're looking at. It's really important to know all of the medications that the particular donor is on as well. All of those things, even though they end up being more surveys and more time for caregivers and family members to complete, are really critical to be able to interpret some of the biological information that's coming out from these biosamples. Okay, I will skip the other parts for right now. So for me, I think one of the biggest takeaways is that, there are a lot of places in the drug development pipeline that I showed that require participation involvement or community involvement. So whether that is doing those, online interviews to do disease concept model interviews, or whether it's to participate in the development of biomarkers through biomarker research. Biomarker research in itself is almost entirely dependent on collecting samples from patients. And so how can people participate in this really important research? So at Combined Brain, we have tried to make it as easy as possible to participate in our research opportunities. And the way that people can donate various blood samples or other biological samples doesn't always have to be blood. We've primarily set it up so that you can donate to the biorepository various biological samples in 3 different ways. One of the first ones is through conference collections. The second one is that we collaborate with clinicians. Most of the clinicians that are seeing these rare or kids with rare disorders have been incredibly phenomenal to work with and are almost always willing to help collaborate with us and, for example, the GRIN2B Foundation to help us collect research samples and research data. And then the third way that we're able to collect samples is just from home visits, having people come to the patient or family's homes. So the first one here that I wanted to talk about is the potential to participate in biomarker research at conference collections. So you can see this table here are the different conferences. So this stands for Hosting Patient Advocacy Group. These are the different conferences that myself or our biorepository team will go to collect various biological samples. Here are the different dates and locations, and then the specific address. Now, the important thing, and this is a little bit outdated here, but is a beautiful figure, so I included it anyway. There are a couple really important things to reiterate here. One, the most important thing is that you'll see the GRIN2B Foundation is not on this list right here. But what this means is that any GRIN2B patients, if they live in any of these physical locations, you are welcome to walk into this conference, even though it isn't hosted by the GRIN2B Foundation, and donate a sample. So if a GRIN2B patient or family lives close to Westminster, Colorado, and they wanted to donate and participate in biomarker research, they could go. They don't have to be affiliated, be diagnosed with CHD2 related disorder. So any GRIN2B participant family who's interested in donating and participating in biomarker research, and you live in any of these cities or close by or could somehow get to them, certainly we welcome your participation. And I will show you specifically how to do that on one of the last slides. The second way, I think, okay, good. The second way to participate in biomarker research is, like I said, in tandem, in collection with clinical visits or clinical procedures. So this is, if you have any clinical visit that has labs that need to be drawn, let's say, for example, your child changed medications and you have to have, sort of monthly, semi-monthly, etc. blood draws. Many times the clinician or the lab will allow us to collect a second tube of blood for research purposes only. We very much appreciate that children typically do not like having blood draws or needles anywhere near them. And so in all cases, we make every effort to try and reduce the amount of needle pokes or times that a kid has to go through that potentially traumatic experience. It is certainly not limited to blood draws. If they're having any other types of clinical procedures and there are supplemental samples or anything that's stored left over, we can collect it. You saw on one of the last slides, there are lots of different sample types that researchers can use to do some of these biomarker studies. So whether that is collecting a second vial of blood, whether that is there was a biopsy that was required to be taken or a tissue that was resected for some purpose, anything that is not needed clinically could potentially be stored and saved for research purposes. And then obviously cerebral spinal fluid, if there is ever a case where your child has to have a lumbar puncture or otherwise open the space where cerebral spinal fluid is accessible. Cerebral spinal fluid would be something that many researchers could potentially leverage and use to look for biomarkers for GRIN2B related. Okay. So probably one of the most important or most used ways to participate in biomarker research is home visits. So this is just as the name suggests, we can collect a variety of non-invasive biological samples through coordinating technicians or other sort of medical assistants, phlebotomists, nurses, to come to the patient or family's home to collect a variety of different samples from them. Lots of these samples don't even require any type of medical assistance. For example, nasal brushings or buccal swabs that are relatively non-invasive and can be completed by other family members. So this is just to remind me to say sort of the process for this is, we send a variety of collection kits with the necessary reagents, instruments, tools, etc., tubes to the individual's home. We organize various medical assistants or professionals to come to the participant's home, and those samples can be collected and stored for GRIN2B researchers. Okay, so one of the most important questions and something that I always want to make sure I highlight is who exactly should participate in biomarker research? So most people are probably very aware that samples from affected participants are incredibly important. But one thing that I think goes often unappreciated is that samples from unaffected participants are almost equally as valuable and important. And these would be, siblings or even parents in some cases can be really good control samples for affected participants. If you think about it, it's really difficult to identify unique differences in an affected population without having an unaffected population to compare it to. So always remember that affected samples are very, very important, but just as important are getting samples from unaffected, potentially age-matched, even sex-matched, unaffected neurotypical participants as well. Here's a picture of one of my favorite families. This little kiddo in the middle is diagnosed with something called Coulant-de-Vries syndrome, but a picture here to show that like participation in biomarker research for the entire family is super important. So all ages, races, and ethnicities. Also, just as important is you want all severities to be able to capture the broadest clinical spectrum of disease. So you want samples from patients who have a very mild clinical presentation to very severe clinical presentation. And that is because you want to identify biomarkers that are relevant and appropriate for the broadest clinical phenotype for what GRIN2B related disorder can potentially look like. Okay. How to participate in biomarker research, part 2. So this is actually just sort of the nitty gritty of how you would go about this if I haven't dissuaded people with needle talk and lumbar puncture talk already. So the way that you would go about this really is the first step is really reaching out to somebody at the foundation, the GRIN2B Foundation, and say, hey, I'm interested in participating in biomarker research. And a lot of times this can be before you have a clinical procedure. If you have something scheduled and you think that there might be an opportunity to collect anything that is residual or supplementary or leftover, et cetera, this would be a really good time to contact the foundation. It does take a couple weeks or some time to sort of coordinate, but this would be a really great opportunity to leverage and put samples in the biorepository for GRIN2B. The next thing that would happen after you contact the foundation is that someone from Combined Brain, our staff, will contact you and consent to participating in biomarker research. What this looks like is some video teleconference, Zoom, Google Meets. It usually takes somewhere between 30 to 45 minutes. The time variable here really is the number of questions that are asked by families, caregivers, patients, etc. One really important thing is that we need genetic reports and able to be able to understand the types of samples and the types of information that we're collecting from our biological samples. So actually one segue here that I wanted to just pause is that my colleague, Dr. Weston, actually put together this super great overview of how to read your genetic report, because sometimes people get their genetic report and sometimes people just know what their genetic variant is, but I thought this would be potentially a really helpful segue. Especially since she already put together this slide. So this is an example of a GeneDx genetic report. Not everybody's is going to be through GeneDx, but the layout for these genetic reports typically tend to be very similar. So the type of test that was requested, some people here want to know whether it was done through whole exome sequencing or whole genome sequencing, or whether it was a specific gene panel just to look for genes associated with epilepsy. That type of information is usually collected here. So if someone asks you what type of genetic testing, that is where this information usually is written. The second one is a clinical indication, which is why did the doctor order the genetic testing to begin with? It doesn't necessarily have to be epilepsy or seizures, but many times, for example, for a GRIN2B related disorder, this probably will be the reason. And then the more important thing, and one of the things that's really important for us when we're collecting samples from patients, is to know and to confirm that they in fact do have a GRIN2B variant. So here I've highlighted in yellow here, these are just examples, not real genes, but here you would have GRIN2B. You can see the amino acid variant, and then you can see the cDNA region, the change in cDNA that is associated with the particular GRIN2B genetic change. And then over on the far right side, in this case, this one is a variant of unknown significance, but in many cases, for people who are diagnosed with GRIN2B related disorder, if GRIN2B is listed here in the genetic report, the classification will in most cases, will probably say pathogenic. And then a short explanation of where it is and sort of the clinical symptoms that are associated with any particular diagnostic genetic result. Okay, so that is just to help people. These two pieces of information are probably the most important. You might also have additional genetic hits or results in this table here. Those are also really important to know as well. So I'll go back. When you're consenting with someone from our staff, it's super important to either know this information or preferably to have it on hand so that we can see it and confirm. Also a list of medications the participant on, and then there's a variety of neurodevelopmental surveys that we request caregivers to complete. So Combined Brain will then help coordinate the sample collection depending on the particular method that we're collecting samples, whether it's a conference or whether it's with a clinician or whether it's a home visit. There are a number of questions that are asked immediately before or right after sample collection. We collect the sample, we process it, and then we store it on behalf of the GRIN2B Foundation. So the GRIN2B Foundation is the person or entity that gets to decide who has access to their samples, which researchers are able to receive those samples. One really important thing is that we have an online catalog that shows researchers worldwide what samples we have. It is de-identified, so it doesn't have your child or adult's or adolescent's name attached to it. But we do want to distribute these samples to as many as people as possible. And so we do put this information about sample types, genetic variant on our website. But again, it's in a de-identified way. But this is in our effort to sort of expedite the development of treatments and biomarkers for rare diseases. And then the last thing that I'll say is that participation outside the US is possible. It just requires sort of the coordination and facilitation with either a researcher has their own local IRB or own local ethical approval to do clinical research that includes biological sample collection, or a clinician who is willing to help us collect research samples abroad. But this is possible. And so people, if you're watching this from outside the US, please still reach out if you're interested in participating in biomarker research. Okay, so I think I did it. This is my last slide. That was a little bit longer and wordier than I had meant, but there you have it. I guess I'm good at talking. Okay.
That was wonderful.
Yeah.
So I don't see any questions in the chat, but I am enabling the option for folks to unmute themselves so that they can ask questions if they would like to. But we have a question for you from GRIN2B. So I know some of the families that are probably watching this have given blood samples to Searchlight at past conferences, at past GRIN2B Foundation conferences. Is that collection different from the collection that you all are doing? Like, are the samples collected for the same purpose? Or if these families gave a blood sample to Searchlight, should they participate again?
Yeah, that's a great question. So, there's a couple of answers to that. The first one is that we collaborate really closely with Simon Searchlight. They are an amazing organization that has been doing this way longer than we have. And so the first thing I should say is that whenever possible, when Simon Searchlight and a team from Combined Brain are at the same conference, we collaborate together. And so we collect samples at the same time so that participating in both does not require additional needles and pokes and stuff like that. So that's the most important thing we figured out first. The second thing is equally as important, is that we have intentionally designed what we collect to be different from what Simon's Searchlight collects. So Simon's Searchlight collects still blood samples, but they store them and process them very differently, and they use them for different purposes than what we collect our tube of blood for. For example, one of the main things, and I hope I'm not misspeaking, but one of the main things that Simon's Searchlight does when they collect tubes of blood is they isolate different types of white blood cells. So white blood cells that they can potentially just store and freeze until they figure out what they want to do with, or they reprogram those and they make them induced pluripotent stem cells, which are cell models that I'm sure lots of people have heard of. And those are incredible resources for researchers to use. They also isolate DNA from a small volume of blood that they collect, which is also super important for researchers that are looking at sort of genome-wide variants or even epigenetic changes. So that's what they primarily focus on. We can do all of those things as well, but in terms of coming up with biomarkers and collecting materials that optimize sort of the sample types that we can use to do sort of omics biomarker screens, and to not overlap and collect the same things that Simon's does, is we collect and process things for things like plasma or serum. And those allow us to look for certain proteins or metabolites or mRNAs or other sort of molecular indicators that could be biomarkers. The third part of your question, I realize this is the world's longestly answered question ever, but the third thing is that the main thing about a biomarker is it changes with age, or it changes with disease status or progression. So theoretically, you should collect it maybe once a year, or every other year. Or, we know that collecting blood from kids is not the funnest thing in the entire world. But if you're looking at things and trying to make sure, yep, they do change with age, you gotta collect multiple times to make sure it does actually change as the kid gets older, or as the adolescent becomes an adult, or etc. So yes, you should absolutely, if you have donated through Combined Brain or you've donated with Simon's, you should donate as often as you feasibly can.
Awesome. Yeah, I know Grin2B Foundation was not on that list of locations for conferences for this year. We host our conference every other year. So our last one was last year, 2024, and we're hosting again next year. So I know some of our families are maybe waiting until next year's conference to do those samples. But like you said, if any of our families are located within a close proximity to the locations that you have listed on that tour schedule, please make sure that if you are able to, that you can go out and get those samples taken care of. So that was a great question. And then one other question that I had, and you briefly went over it, but I just wanted to re-ask the question for clarity.
Yeah.
So any researcher can request the samples. I know you mentioned that there was an interest in getting the samples distributed as internationally as possible. So these samples will not necessarily have to wait for Grin2B Foundation to send over a list of, these are the researchers we want to receive these samples. Any researcher can access them with the protocols that you guys have in place at any time.
Yeah. So we have a catalog online of like all of the resources and samples that we have. And so we are usually contacted. We usually distribute samples in a variety of ways. The first one is like researchers find us through our website. They go to our website, they see we have these things, and then they place a request through our website. And then the other way that we usually get a hold of scientists and give them samples is that they contact you, or they go directly to the foundation. They go, Alyssa, hey, I found this thing that I think could cure Grin TB kids. Do you have some, a cell line, or do you have some plasma samples I could potentially test it on, or look at? And then you come to us and you go, okay, Dr. So and so needs this. Can you ship him this? That sort of thing. The critical thing, though, is that we hold, for example, Grin TB. If Grin TB had samples in our repository, which you guys have a few, we don't send anything out until you tell us to, right? So whichever way they contact us, whether they come through our website or whether they go through you, before we send anything out, we have to go, okay, Alyssa, are you want to give Dr. So-and-so these samples? You say yes, we send them. That's how that works.
Awesome. Thank you so much for that clarity, because I was paying attention, and I said, let me ask, because I know someone else out there probably has
the
same question that I do. So that is wonderful.
The other thing that I want to reiterate, just to make sure if there's anybody listening, we give them to whoever you tell us to send them to. So that could be an academic, that could be an industry person, that could be another nonprofit, etc. And so, most cases, I mean, it will be all of those, right? We're trying to promote the increase in research in rare diseases, and so we don't say no to people unless you tell us to say no to people. So when people donate, they have to keep that in mind. That could go to anyone.
Got it. And that's great, that ever needing sense of all of these nonprofits doing the same work, or industries and researchers and scientists doing the same work, and no one knows what the other hand is doing. So I think that opens the door for great collaboration, and really fast tracks how we access diagnoses, treatments, cures. So I think that's wonderful that you guys make sure to send whatever is needed to whoever across the board. So I think that's great. Awesome.
Cool.
Well, Anna, I think taking a look here, and I'm looking, and I was like, in the interest of time, that was a wonderful presentation.
Okay.
I am surprised that we have ended before the hour, but thank you so much for presenting today. I hope everyone who is watching or listening gains great value from Dr. Falzer's presentation. And if you have any questions, I know it was mentioned for you all to reach out to the foundation prior to participating. Please reach out to us at either the info@grintb.com email address, or to Liz's email, which is liz.marfia-ash@grintb.com. Just feel free to reach out to us with any questions. Even if you have some for Dr. Falzer, we will relay them over to her and the team at Combined Brain. But thank you all for participating today and joining us during Grin TB Awareness Week. We really appreciate it, and we hope to see you guys again soon. All right, Anna, thank you so much.
Oh, you're so welcome. I'm so glad I could do this.
Definitely.
Great. Yay, Grin TB Awareness Week.
Thank you.
You're welcome. See you later, Alyssa.
Bye-bye.